Welcome to my second video. If you have any question about a previous video, please post questions on Edmodo, or I don't know, write down in the comments, because those videos are going to be posted on YouTube. Right now, we're going to continue with the same lesson, Mendelian genetics. The previous video covered the first 2 pages. Now we're moved on the page 254. We're going to explain the key terms. So our first term is allele. So what basically allele is allele is an alternative form of a single gene, or we can say it's a different form of one or single gene. So what it means, if we have a gene that determines our eye color, alleles are going to be different forms of that gene. So basically one allele is going to determine the blue eye color, the second one is going to determine brown, or I don't know, black, green eye color. So basically it's an alternative form. Alleles are alternative form of single genes. So maybe you recognize this picture. It represents what? Yeah, homologous chromosomes, or pair of chromosomes. One come from father, one come from mother. As you can see here, I pointed out the gene on a specific place on both of those chromosomes. One gene, that gene determined the eye color. For example, this one that comes from father determined red eye color, some red eye color, and this one blue eye color. So basically, this can be in Mendelian experiment. This one can determine the yellow color of the seeds. This one can determine, for example, the green color of the seed. So in the previous video you saw that in F1 or first filial generation, there is only the seeds with yellow color. So those with green color disappeared, although one of the parental plant have a green seeds. But in first filial those green seeds disappeared. But later in the second filial, once again the green seeds appeared, but the ratio is 3:1. So on every 3 yellow seeds there is 1 green seeds. So basically Mendel concluded that that ratio 3:1 could only be explained if alleles were paired. So what basically that means? That means that the form that appeared in the F1 generation is dominant form, or that allele can mask the presence of another allele. So let us say this one, the F1 generation got both one yellow color, one green color, but the yellow color is dominant, so he can that form can mask another form. So the another form is recessive. So basically that means when it's come in pair with dominant, the dominant is going to be shown. You can see only dominant form. Okay, the second 2 terms that we're going to explain are homozygotes and heterozygotes. So basically what it means, an organism with 2 of the same allele is a particularly homozygote. So homo means basically the same. So if F1 or F2 generation, I don't know, the offspring get from parent the same allele, they will be homozygote. If they inherit the different, one for yellow, one for green, it's going to be heterozygotes. 2 terms that also another 2 terms that we're going to explain are phenotype and genotypes. So phenotype is basically what you can observe, what you can see. So you can see green or yellow color. But genotype is what determined those those green and yellow colors. So an organism allele pair is called a genome. So genome you cannot see, phenotype you can see. So phenotype is going to be black hair, brown eyes, white skin, something that you can see, and genotype is something that's in your genes that you cannot see. Okay, now we are going to move on the Mendel's laws, so be sure to watch the next video. Thanks. Okay, those findings Mendel used to develop a law of segregation and the law of independent assortment. So we're going to explain first the law of segregation. So what basically that means? That means that two alleles for each trait separate during meiosis. Your textbooks, you can follow this page 255 or this diagram here. So basically you can see here yellow and green seeds. So this one are homozygotes for green and yellow color. Or okay, I can use this one. So basically this one for yellow, this one for green colors, homozygote. Basically the first law states that alleles separate. So this one separate from this one separate from this one. That will result in an offspring that are heterozygotes, or we call them hybrids, with two different alleles. Okay, now we're going to move on the second law. Second law of independent assortment. It states that random distribution of alleles occurs during gamete formation. So what's the gamete? Please try to remember. Pause the video now. Okay. If you remember or you did not remember, the gamete are reproductive cells that have haploid number of chromosomes. You can pause it once again to check what's the haploid number of chromosomes. Okay, but now we're just going to continue. I will try to explain. So in your textbooks, you can see this picture here. This is kind of like a cell, and those are the possible allele combination in the gametes. So this one, capital Y, represent a yellow color. This one, lowercase, represent green color. This one represent capital R, represent round shape, and lowercase r represent wrinkled shape. So basically the random which one gametes you will get, it's a pure coincidence. Are you going to get the all capitals, one capital, one lowercase? This is genotypes inside. Phenotypes are what you can see. So round yellow shape, round yellow colored, the wrinkled yellow color, or green round, or green and wrinkled. So basically it's random distribution of gametes, of alleles during gamete formation. Now I'm going to explain the Punnett square. So basically in F1 generation, as you know, I hope so, that all of the seeds are yellow. We explained that is because yellow allele is dominant over the green one. So basically you can see a hybrid female and a male one that have two alleles. One for, so capital is for yellow color, the lowercase is for green color, but because the yellow color allele is present, it hides the green one. That's F1 generation. That's why you have all yellow. If you cross between those hybrids, in F2 generation you will get the ratio 3:1, or 3 yellow, 1 green. So basically if you separate each allele here, form like a table, so Y, capital and lowercase, capital, lowercase, and you combine them, capitals, then capital with lowercase, lowercase with capital and lowercase with lowercase. You will get the genotypes 2 capital Y, one capital Y. So this one will be yellow. These two in the middle are going to be also yellow because the yellow color is the dominant, and this one will be green because there is no yellow that's going to mask that green color. There are only 2 green alleles. So ratio of genotype is going to be 1 to 1, but the ratio of phenotype is going to be 3 versus 1. Okay, now we're going to move on dihybrid cross. Finally, we'll finish this lesson. Okay, dihybrid cross basically follows the 2 traits. So far we followed just one trait, for example, green and the yellow color. Now in dihybrid cross, our Mendel tracked down the 2 traits like seed color and the seed shape. So there are 2 types of the seed shape, round one and wrinkled one. So basically when he cross between those plants that have that are homozygote for yellow and round shape and green and wrinkled shape, he get in F2 generation ratio 9:3:3:1. Basically that means in F2 generation he got first in F1 generation he got all yellow and round shaped. This is parental gen generation. There is no, as you can see, green or the wrinkled one, but here in F2 generation there are 4 green and 12 yellow. So if you look in your book, you will see some of the yellows are wrinkled. So basically the ratio is 9:3:3:1. So 9 yellow and round, then 3 green and round, then 3 yellow and wrinkled, and only 1 green and wrinkled. Okay, and just to mention probability. So basically the probability, if you trace only one trait, like seed color, you will get 3 against 1. So the probability to be offspring, for offspring to have a yellow color is 75% versus 25 for green one. Okay, thanks. We have another lesson. It's going to be a lot shorter. Hopefully it will fit in one video. So make sure to watch it. Bye bye.